Good morning. My name is Khawla El-Rafi'i. I am a Senior Research Associate at the Qatar Biomedical Research Institute. And it is with a great pleasure that I welcome you all to our web seminar titled Defining Key Elements in a Research Institute QBRI Core Facilities. This web seminar will be featuring individual core facilities in QBRI and relevant technologies. First, please allow me to introduce Qatar Biomedical Research Institute. QBRI is a national research institute under Hamad bin Khalifa University and part of Qatar Foundation's Research Development and Innovation. QBRI is a disease-focused research institute engaged in basic and applied medical research that strongly supports the translation of novel scientific discoveries into better preventive strategies. The spotlight of QBRI is on applied research, concentrating on applying integrative and multidisciplinary approaches toward associated health challenges. The Institute simultaneously focuses on tackling more effective means of diagnosis and treatment to develop applications that will improve the quality of life and effectiveness of healthcare in Qatar and the region. In keeping with Qatar National Health Priorities and Qatar National Research Strategy, QBRI focuses on research areas related to diabetes, neurological disorders, cancer, and recently infectious diseases. During this web seminar, QBRI Core Manager will be demonstrating the state-of-the-art services QBRI labs can provide to local and international entities and help them increase efficiency. All our core facilities are located in the same building, a feature that provides easier integration of multiple services. So before we start with our speakers, I would like to mention a couple of housekeeping rules. The microphone of all attendees have been muted, but we encourage you all to post all the questions and during the talk in the Q&A tab. Also, please direct this question to all panelists to make sure your questions are visible to us, and make sure to mention the core you are addressing the questions to. Unfortunately, Dr. Lawrence could not be with us today, so without further ado, I would like to welcome our first guest speaker, Dr. Gerald Pfister, the manager of Flow Cytometry Core. Dr. Gerald, I will hand over to you for your presentation. Can you unmute yourself, Dr. Gerald?
So, hello. Can you hear me now? Thank you very much, Paolo, for this kind introduction. I will start with my presentation immediately now. So I'm sharing. I hope everybody can see the slide now. And I will start without any further delay to present the flow cytometry core in the context of the QBRI core facilities. So in this presentation, I want to give you a brief overview of what the flow cytometry core can offer to researchers in terms of instrumentation, of infrastructure, and of services. Before we start with that, I want to give you a very brief overview of what flow cytometry in general can offer to biomedical research and biological research in general. Some of you might not be so familiar with flow cytometry. So very quickly, what flow cytometry basically can do is you pass individual cells from any kind of material, if it's blood or tissue or water or whatsoever, through a flow cytometer, and it passes some laser beams and optics there, and then you can analyze individual cells by flow cytometry, and you can sort individual cells by cell sorting. The big advantage of this methodology is that it's a very quick and very accurate methodology. So it's easy to pass thousands of cells per second through those machines and to sort the same amount of cells in this time. Now, a few fields here where flow cytometry plays a big role. It's immunology. That's a field of flow cytometry as such. If it's immunophenotyping or cell signaling, most of you might know that. Cell biology or cancer research is another big field where you can analyze things like apoptosis.
Dr. Gerard, sorry to stop you here. We cannot see your presentation, please.
You cannot see my presentation. Okay, wait, sorry, why can't you not see that? I'm very sorry. I need to go out and we'll start again. Can you see now?
Yes, it's good now.
Sorry, something went wrong before. Okay, so that's what I was explaining before. So very quickly, that's the principle of flow cytometry, where the cells pass the laser beam and you can analyze thousands of cells in a few seconds. Then the main fields, flow cytometry is useful, very useful in research, is immunology, cancer research, cell biology, for instance. That's what I mentioned before. If it's apoptosis, if it's immunophenotyping, if it's calcium flux, things like that. Another field where flow cytometry is really an helpful tool is fluorescent protein analysis. So you can easily detect expression of fluorescent proteins like GFP, YFP, and so on with flow cytometry. Plant biology, that's not done at the moment here, but that's where you could look at DNA content. Of course, you can do the same in animal cells or human cells as well. Microbiology, that's another field where flow cytometry is more and more important to look for bacterial viability, bacterial activity, microbiopsies in milk, in nutrition, in water, and so on. And of course, also stem cell biology. Again, for immunophenotyping, flow cytometry is really useful, or for other things like cell signaling. That's just to give a quick overview of what can be done with flow cytometry in our context here. Now, I lost. Okay, now here in this slide, I want to give you a quick overview what the flow cytometry core here at QBRI can do for you and what is our mission. We are providing efficient and reliable flow cytometry and cell sorting support and solutions with highest standards of instruments, quality control, also biosafety and productivity. We give access to state-of-the-art installations in our core, and we provide professional services for flow cytometry analysis and cell sorting, and we have the skills and expertise to do so. Now, this is an overview of skills that we have to offer in our flow core. This reaches really from the beginning, like sample preparation, how to prepare the sample for flow cytometry, how to design a panel for flow cytometry, do multicolor flow cytometry, how to combine different colors in flow cytometry, over to sample acquisition, how to acquire best samples, how to analyze your data. Very specific field would be compensation, optimization of panels, and development of panels as well. Of course, we can do cell sorting in single cell assays or in bulk sorting assays. And also we do offer education and training on our instruments, on software, on assays. So this is basically the big field of skills and expertise we can provide. Now, to show you a little bit what infrastructure we have in the core, I want to start with the flow analyzers we do have and we can give access to. We have up here, that is our workhorse. It's the BD LSR Fortessa X20. It's a SOR, special order research product flow cytometer that can analyze up to 20 different colors, and it has 5 different lasers in it. With this machine, you can basically see any conventional fluorochrome and color that is available at the moment. A special thing we have here, it's this box on this machine. It's called an HTS, high throughput sampler. If you would have to analyze many samples, you can analyze directly from a 96 well plates here, for instance. Then down here, this red one, it's a Akuri C6 flow cytometer. That's a very simple and easy to handle flow cytometer that is used also routinely in our labs here, that can be used for quite simple assays, but it also can do some things the bigger one cannot do. For instance, with this machine, you can do a direct count. If you are interested in how many cells per microliter blood you have, you can do a direct count with that machine, for instance. Another analyzer is the Gallios with 3 lasers and 12 detectors as well. A specific analyzer would be this machine here. It's the Amnis image stream. I will show you in a slide a bit later what this specifically does, but in fact, this is a nice combination between flow cytometer and a microscope. So it does both imaging and flow cytometry. Another very specific machine we do have, it's the CellC, CTC single cell analyzer. What this machine can do, it can analyze circulating tumor cells in very low concentrations, blood, for instance, just by size. The tumor cells are captured on a microchip here, and then you can analyze those cells. The fascinating thing with this machine is it can detect something like 5 tumor cells in 1 milliliter of blood, so it's very sensitive. Further on, cell sorters, we have 2 big fax area cell sorters with 5 lasers, and again, 18 colors can be analyzed with those cell sorters. And we have one BDFAX Jazz, which is this one here, that is located in a biosafety level 2 cabinet, as you can see here. So this can be used for more tricky sorts in terms of biosafety. Now, all our sorters can sort bulk sorting. So here you see the fax area cell sorters can do 4 populations sorting at once, where you would see here 4 collection tubes, 4 populations separated and sorted individually. What we can also do is sort into a device like a 96 well plate. So for instance, we can here do single cell sorting, and we can sort 1 cell in each well and start a clone culture, for instance. SORT, again, stands for Special Order Research Product. What that means in terms of our sorters is basically that we can have, first we have 5 lasers, which is really a lot, and we can individually tune those lasers in terms of power. So that's useful when you want to look at very sensitive cells that might be damaged by too high laser power. So we can adjust this to the sample specifically. Now, going on with the general lab infrastructure we have dedicated analyzer and sorter labs with controlled user access, which is important in terms of biosafety. We have online booking system where you can book in and book a service, for instance. That's something Adel will explain more in detail at the end of the session. We also have a fluorescence microscope to check for cells, for fluorescent cells. And we also have a software analysis station. So the common flow cytometry software would be FAXDiva, FlowJo, Sortware, for instance. We have all of this available to do analysis of the data here in our labs. And of course, general labware and centrifuges and some things like that we can also provide. Now, going into the services, what services do we offer as a flow core? We give advice in sample preparation, as said already, reagent selection, protocol design, optimization of protocols. We give advice and support in multicolor panel designs with controls, what controls are needed, what compensation has to be done. Multicolor sample acquisition and analysis is done. Single cell analysis and sorting applications. As said, we can do sorting into a collection tube as a bulk sort. We can do single cell sort in 96 well plates, for instance. Biosafety is a big issue in cell sorting as well. What we do routinely is surface phenotyping or intracellular staining, apoptosis assays, cell cycle analysis, proliferation analysis. Those are all things that can be nicely done by flow cytometry and in our labs. And of course, we provide training and education as well, in terms of university courses, but also on an individual request if you want to get some educational training specifically tailored for your needs. Now, all these services come together quite nicely, and I showed this slide here because this is what makes a successful flow cytometry experiment at the end, from beginning to the end. And our services basically cover all those steps, from experiment design sample preparation, fixation, controls, over to instrument and settings, select the right instrument, which lasers, which filters do I need, what instrument settings are required. Then go to data analysis. Interpretation of data can be sometimes tricky in flow cytometry, so we help with that. We help with gating, how to display the data, and then also, the display data, how do they look best and how are they displayed correctly. Those are all fields that can be easily covered, and we do so in the flow cytometry core service. Now, I want to show you a few selected activities that display a bit what I mean with all those services. So this is something we do routinely in our lab. It would be a 4-way sorting of different T-cell populations on our cell sorter, Faxaria cell sorter here. So you have here all the populations of blood cells. We do doublet discrimination by gating, shown here. We gate for only live cells. That is done by a fluorescent dye called 7AAD in this example. So only the negative cells are live cells for that, and they are selected. And then we can define, according to the staining, different cell populations. In this case, it's CD4 T cells, regulatory T cells, effector T cells, and those cells can then be sorted. And as a result, you get very pure, so this, for instance, orange population here is after sort, very pure, nothing else is there. Regulatory T cells, pure population of those after the sort, and the same with those effector T cells. So you get pure population of cells after we have done the sorting. Another example would be small particles. In this case, it's nanoparticles, 100 nanometers, the smallest one, can be sorted with our instruments very purely and very nicely. As a real-life example, also, I added here an assay that was performed by Dr. Young-Soo Park from the Neurodegenerative Research Center. So those were exosomal vesicles that are very small, below 100 nanometers. And here you see background in gray, and the EVs would be the blue ones here. And you can analyze them when you stain them even better, and you can also sort those. So this is a little bit a tricky setup because those particles are very small, but it works very well with our machines. This is this imaging flow cytometer I showed you. Just to briefly go through, you have a light imaging. Yes, time is over soon, I know.
No, all right, thank you.
Thank you. I will do one more minute, please. 1 minute. Right. Okay, so on this machine, you have a bright field image as a microscope, and then you have different fluorescence channels where you can see, again, for instance, different surface markers stained, and you can see morphology of cells and the size of cells as well. Now, the last 2 slides show recent highlights of our research, and this brings a bit into context what where flow cytometry lab comes into account in a whole research paper or research work that was done together with the cancer research team. What they did is we sorted out of PBMCs specific T cells, and then they grew from these sorted cells another specific population of regulatory T cells that play a role in cancer development. So what we did is we did the cell sort, we got pure populations. Then for the analysis of the cells, flow analysis was done. A specific type of analysis was done down here. It's a proliferation assay. So each of these peaks would represent one generation of divided cells. So you can follow up in your cell culture how many cells divide, what generation, how many generations you have, and you can follow up individual generation of those cells, again, further with flow cytometry. The last slide now, another publication that is very recent. It just was published, in fact, right now, December. And this is a test we did here in the flow cytometry lab. Often you hear people concerning about cell sorting destroys cell or reduces viability. So we took our sorters and did cell sorts on different sorters up here and sort conditions, and we checked the cell viability of sorted cells compared to NS, means not sorted cells. basic outcome is sorting does not kill the cells, so the viability was high and not changed towards not sorted cells in our experiments. Also down here, proliferation worked well after cell sorting with different cell sorters and conditions as compared to the not sorted cells. So what we can say confidently is that our cell sorting does not kill the cells, but not influence the cell proliferation. So we are happy to provide good quality cells after the sorting. And with this, I want to thank you. And if you have any questions, please let me know via email or at the end of the session, there is a Q&A session where you can also address your sessions. Thank you very much.
Thank you. Thank you, Dr. Gerald, for this informative presentation on the flow cytometry core and its relevant technologies. And now our next speaker is Dr. Khalid Warghini. He's the core manager of the genomic in QBRI. And now, Dr. Khalid, the floor is yours.
Yes, hello everyone, and thank you, Khawla, for your introduction, and thank you all for joining us, and welcome to this core webinar. So as mentioned, my name is Khalid Warghini, and I am core manager of genomic facility. So this presentation, it will be on two parts. The first part of 10 minutes, it will be devoted to general overview of our facility. And the second part, which will be presented by my colleague Richard, and it will be focused on bioinformatics analysis services. So, as you know, the genomics is one of the most exciting and fruitful area of biological research of this 21st century. So, in our facility, we have different platforms and we offer different services to support different aspects of genomics project, from sample processing to data generation and analysis. So our services are next generation sequencing, single cell sequencing, microarray services, and single sequencing. So for next generation sequencing or NGS services, for this service, we offer a complete service from sample processing to data generation and analysis. So after sample submission, the quantification and quality control will be checked. Quantification will check by using qubit or nanodrop. And then for quality control, it will be checked by using nanodrop for DNA and by using bioanalyzer or lab chip for RNA. So once the quality control has been checked, we'll proceed with library preparation. For library preparation, we offer different library preparation for different applications, like for RNA-seq, implicon sequencing, whole genome sequencing, and exome sequencing. So the library preparation can be manually or automatically by using Agilent Bravo NGS robot. And this depends on number of samples and type of library. So and after library preparation, the quality control will be checked, and then the library will be sequenced on Illumina sequencer platform. So as platforms that we have as a sequencer, we have two high throughput systems. We have HiSeq 4000 system, and we have HiSeq 2500 system. And as low throughput system, we have MiSeq. So HiSeq 4000 system can sequence up to 96 exome, or up to 12 human genomes, 100 transcriptomes, or up to 192 mRNA. So HiSeq 2500 system can sequence up to 72 exomes, 80 transcriptomes, or up to 150 mRNA. So for MiSeq, we can use it for to sequence small genomes or target genomic or target RNA. So once the sequencing is completed, the quality control of the run will be checked. And then the data, the raw data, will be transferred in FASTA format to the user with a complete report of service. This report contains an overview about the service. It means the samples that you use here. And this example is total RNA, and type of library. Here, this example is total RNA, and sequencing, where we described which machine that you used. Here is HiSeq 4000, and number of reads that we want for each sample. Here is 50 million reads, and also contains the quality control of different steps of service, quality control of the samples, quality control of library, and also quality control of the run. And it contains also FASTA report, and FASTA report which contains number of read of each sample and Q30. So the next service is single cell sequencing. This service is to explore the transcriptome at the resolution of a single cell. This has emerged as powerful set of technology for studying rare cell types, individual cellular heterogeneity, individual cellular signaling pathways, cellular ecosystems of tumors, individual cell differentiation, or assessment of drug resistance. So here is the workflow of the single cell servicing. For this service, we request single cell suspension on ice with extra volume suspension buffer. The cells can be from different origins, from tissue or cell lines. And once we receive the cells, the viability QC will be checked. For that, we request 95% cell viability. And then the cells will be isolated by using DDSEC machine. DDSEC system is a droplet-based microfluidic system and can isolate up to 1,200 cells and capped only 3' mRNA, and can support up to 50 micron cell size and incorporates UME code. So once the cells were isolated, we'll proceed with library preparation and then sequencing on Illumina platform, and then generation of data. And data will be shared in FASTA format as for NGS with a complete report. So the next service is microarray service. For microarray service, we have a broad range of microarrays, including transcriptomes microarray, genomics microarray, and epigenetics microarray. So for transcriptomics microarray, we offer gene expression profiling, microRNA expression profiling, and for genomics microarray, we offer SNP profiling and copy number variation. And so for epigenetics microarray, we offer DNA methylation profiling. So for that, we have two systems. We have Encounter from NanoString, and we have iScan from Illumina. So here is an example of workflow for Encounter gene expression. So from RNA, extracted RNA can be from different origin, and this for Encounter can use also RNA extracted from FFP samples. So this RNA will be subjected to hybridization by using two probes, capture probes and reporter probes, and then purification by removing excess of probes, and then the cartridge will be read on Encounter system, and data it will be on CSV format. The second system is iScan system. Here is an example of Infinium assay workflow. Is a SNP profiling. So the genomic DNA, it will be amplified, fragmented, and then precipitated and subjected to hybridization to the bit shape. And there is extinction and staining, and then the bit shape, it will be imaged by iScan system. So the next service is Sanger sequencing. We have 3 machines. We have 96 capillary machine, and we have 24 capillary, and we have 16 capillary. So for this service, we accept different templates. The most popular is purified product like plasmid and amplicons. Sorry, and we also accept a non-purified product like PCR product. We have your template primers and all mixed, or directly colony by using rolling circle amplification. So here is the genomics team. Here is Dr. Richard, who is in charge of bioinformatics, and he will present the second part of this presentation about bioinformatics services. Ms. Khawla, she's with us today. So she's in charge of NGS services, and Ms. Sumaya, she's in charge of Sanger sequencing services and microarray services. Thank you.
Thank you, Dr. Khalid, for this broad overview on the genomic core in QBRI. And now joining Dr. Khalid, Dr. Richard, our bioinformatics specialist. He will be presenting the bioinformatics support provided for the genomic core. Dr. Richard, I will hand over to you.
Hi, thank you. I don't seem to have that. There we go. There we go. So I'm just going to very quickly overview the bioinformatics that we have at QBRI, just to give you a feel of some of the work that we're doing. So our primary platform for the bioinformatics is our cluster, which is made up of 472 cores spread across 10 36 core nodes and 2 56 core fat nodes. The way these clusters work is that you treat it as a whole computer. And so there's a central head node that distributes the jobs, allowing you to run multiple jobs either across more cores than you would have in a normal server, or running multiple jobs at the same time. So this is our primary platform for the work, but we have other servers available, so there's lots of other capability. So our primary work is through the genomics core, primarily the whole exome sequencing that we do through so once the sequences have been done by Khalid and Hala, they are processed from the proprietary Illumina format into the FASTQ standard format, and these files are then aligned. For whole exome sequencing, we use BWA, and then we use the GATK pipeline to annotate and call variants and produce usable data for people. We use a similar process for RNA sequencing. Obviously, the BWA and GATK are specialized for DNA samples. So for the RNA sequencing, we use the Tuxedo suite. So here we use Top Hat and Cufflinks to do the calling and annotation. We also have some experience of doing chip-seq analysis. Again, I'm sorry, I apologize. I cannot remember the names of the software we use at the moment, but this is an established pipeline that we've been using with Dr. Ian's work, and it does work very well. Something we've been doing very recently is Hi-C and CIC sequencing. And this is for those who don't know, this is a way of identifying regions of genomic, distal regions of the genome that interact with each other. So for this, we're actually using a Hi-C cup aligner once the DNA has been sequenced, and then we're using this Gothic tool that we've developed in-house in collaboration with CHLS to do the actual analysis and identify the interesting interactions. That is actually all I wanted to mention today. As I said, it was just going to be a very quick overview. If anybody has any questions or anything, they should feel free to contact me. I am open and available if anybody wants training, or if anybody has any inquiries or anything they want to discuss with me, they should feel free to drop me a line. Thank you very much.
Thank you, Dr. Richard, for this brief informative presentation. And now our next speaker is Dr. Prasanna Koltakar, a senior scientist in QBRI and the manager of the structural biology core. Dr. PK?
I am just waiting to get control of the thing. So I'll be talking obviously about the protein. There you go. So I'll be talking about protein, and it says structural biology, but we are a lot more than just structural biology. And as my presentation starts coming up, it was taking a while. Do you guys all see it? Omar?
Not yet.
It's still coming. So it's going to be, we're going to be talking about not just structural biology, but also protein biochemistry and interactions. And so it's much more than structural biology. And how's it looking now?
Can you, yes, just put it on a slideshow.
Can you see it?
We can see it, but if you can put it on the slideshow, because we can see now all the slides.
Okay, that's odd, because I am seeing...
yeah, it's okay now. Yeah.
It's okay now. No, it took a while. The timer. Okay, fantastic. So let's not waste any more time. Everybody can see it. So here's the idea. Most of you in this audience, if not almost all of you besides us, are probably cellular biologists and you have cells. So our core lab helps you to go from these cells to understanding how protein and DNA interact with each other, but also hopefully to try to get a high resolution picture of exactly what is going on. And I'll show you some of the instrumentation and what we can do for you. So in a nutshell, our lab is very different from many of the other labs you could be hearing about today, in the sense that we don't just have 2 or 3 of the similar or same type of instruments. It's a wide variety of instruments. So I've clustered them in a way that makes sense to most of you. So as you can see at the top, we start obviously with protein production. And so here we have 4 or 5 instruments, and I'll go through each of these shortly, that are involved in protein production. Obviously, once you get the proteins produced, you can do various protein characterization, and I'll tell you a little bit about each of these. Then obviously, you have ways of doing various protein and interactions with either DNA, ligands, drugs, for example. And then, of course, the high point is, if you can get to this point, is to do structural biology and get a high resolution 3D structure of something you might be working on. And finally, and this is the only really kind of a standout or standalone, a little bit more exotic equipment, it's called the Nano IR, and I'll tell you a little bit more. And it's not really protein, it does protein and cellular characterization, and I'll tell you more about it. So everything, as I said in the flow that I just showed you, starts out with production of proteins. We primarily work with bacterial hosts. And there we have these large shaker incubator. So we have 3 stacked, and obviously all temperature controlled, and they sit on mats. What's nice about that is these are not obviously steel holders, but mats. So we can accommodate almost, if we can fit inside this room, 2-liter flasks, whatever, it can all be done, and we can make fairly large amounts of bacteria that are then expressing protein. At the same time, if you really want to scale up, we do have a really nice fermenter where you can get to much larger ODs, obviously 10 times the ODs that you would get in kind of a normal bacterial production environment. So there we can get fairly large amounts of protein, as long as obviously your cells and your protein system can handle it. From there, then, I don't want to highlight this too much, but the key is many people might have a Sonicator. The nice thing is this Sonicator has some really fine tips. What that allows you to do is extremely small volumes, which you might be doing to kind of initially test your bacterial expression. Again, I don't like to, this is more for the internal people than the external to QBRI. But the key of this is it's a high-speed centrifuge. But the nice thing is it has many different heads. And I know that can often be a limiting step when you have a nice, beautiful centrifuge, but you don't have enough heads, so we can accommodate extremely wide variety of volumes in this centrifuge. And then obviously for protein production, you see our key unit. This is an Acta Express FPLC unit, and it's a twin, meaning there's 2 of the same, but right, you don't see that in this picture. We have another replica of the exact same thing. So we really have a pretty large amount of a scale for being able to make large amounts of protein. And we do get milligram quantities of protein each week, okay? And having done the protein production, then you might want to characterize your protein. And so we have something called MALS, or multi-angle light scattering. And the SEC, obviously, is the sizing exclusion chromatograph. So that's the HPLC. So in the HPLC, you get something and it's sized differently. And this little box, a very expensive box with one laser, but 18 different detectors, it allows you to get all these other amazing kind of characteristics of your protein, including absolute, not relative, but absolute molecular weight, molecular size, meaning radius of gyration, and of course, even aggregate states. So if some of you who are working on aggregations and oligomers, let's say, this gives you a really beautiful readout of what are your oligomer states in your specific sample. And besides this, then we obviously have a Typhoon scanner, and many people have various scanners to look at gels, for example. This, again, like many other gels scanners, is able to do fluorescent, radiolabeled, now that we have radiation in the lab, and it's extremely robust scans. But what's really important is that they're very quantitatively accurate. What do I mean by that? So we often do mobility shift assays, MSAs, in our lab, and for there, we do KDs that are in the nanomolar kind of range. So we get beautiful KDs in the nanomolar with extremely good reproducibility. So that's the advantage of the scanner. And going back then to protein characterization, of course, have this circular dichroism equipment. Many of you are used to JASCO, but really this applied photophysics equipment is really good. And we had it upgraded last year, so it has a state-of-the-art detector. It makes it even more sensitive. What's nice is that it also has both titration as well as temperature control, so you can do Melton curves. And just recently, we obtained new cuvettes, so you can do fairly small volumes as well to obtain secondary structure information, as well as other characteristics of your protein. Going then from the protein characterization, we have a lot of instruments for molecular interactions. And one of these is the BioCore T200. I will say that my lab right now doesn't have full-scale expertise on these. Like everything else I'm talking about, when I say we have expertise, we have more than 10 to 20, if not even larger amounts of years of expertise. The BioCore T200, we're still kind of feeling our way. We are meant to get even more in-house training, which some of you will be able to avail. But the key is we have all the chips that you would need, all the chemistry that you need to make these slides. And this is to allow essentially label-free understanding of your molecular interactions. Of course, Dr. Omar's lab themselves have some expertise, so we do have expertise within the QBRI. Our key go-to instruments for molecular interactions is the AutoITC. And what's really good about this is that, as the name suggests, AutoITC, it's fully robotized in many ways, including the actual delivery of the sample to the instrument. What's nice about that is it really stops all the introduction of air bubbles that you get with non-automated versions. So it's not about high speed. It's about being able to do reproducible, automatic, bubble-free kind of technology. And it's really our go-to instrument. And we actually have quite a few users, if nowhere else, from Qatar University. So I'm mentioning that because that means that we're not just HBKU or beholden to only what's here in HBKU QBRI. And we have outside users, and we welcome them. And going to crystallization, so obviously once we have enough protein and quantity, we can crystallize things, so we can get crystals. And for that, we have two different ones. This is a very robust but low speed. This is a very, not as robust, but a very high speed. I'm not going to go into those because we only have 15 minutes, but we have both high speed, low speed, robust, all the kind of permutations of crystallization. And I had to bring this up because somebody in the audience will, of course, say, well, we have DeepMind now. We can get structures. Why do we need to have structural biology? And so, yes, we can predict almost any structure now with 90% accuracy. That is true. But as this paper, in fact, said that, it's still tough for protein complexes, protein-protein, protein-DNA complexes. And that's really what biologists want to study. Hardly anybody wants to study a protein by itself, okay? And I'm very aware of this because, in fact, Cas14 is what showed DeepMind. And I have a publication about more than 10 years ago, and that's Cas7. It was about halfway from 90% prediction to, let's say, 40% prediction is what we saw. So I'm very well aware of it. So there's still a role for structural biology, wet lab validation. And so we have a beautiful X-ray machine or X-ray diffractometer with a really good detector, and we also obviously have all the cryo set up so we can freeze the crystals in a very reasonable way, and it's essentially free of liquid nitrogen, so we can do it on the fly. And so finally, as I start concluding, and so Hala doesn't have to cut me off, is that I come to our most exotic piece, which is the nano IR. It's a combination of an atomic force microscope that you see here with an infrared laser. So what's so good about that? So the atomic force microscope gives you a very good morphology of the cell, okay? What's really nice though, and in this case, it's a specific stem cell that we had in the lab. You can drive to each of the different locations of the cell. And this gives an infrared readout, which corresponds to, let's say, protein, lipid, DNA, RNA. So you know exact chemical content of a specific point in a cell. So you can overlay that chemical content on that cellular morphology and create these beautiful maps where in the orange you might have protein content inside the cell, whereas you have some of this lipid in the blue that's kind of enveloping the cell that we're looking at. So, and once again, just like the BioCore, we're still building up our expertise. This is a relatively novel instrument, and we still are building up the expertise. We obviously can do and have done a lot of imaging, and we are still in the process of getting even more in-house training, which we will, again, make available to others. And so in a nutshell, then, in our core lab, we can go from cell biology to not just beautiful 3D structures, but a lot of understanding of the protein-protein DNA interactions, okay? And so currently, what we're here, we help you to collect data, okay? Unless you're extremely well-trained. And when I say well-trained, there's only a few people from Dr. Omar's lab that are allowed to touch these instruments because they're expensive instruments. And if they go broken, it's not just about us. We'd have to blame somebody, okay? And so we only allow extremely trusted users. We do have various in-house training to get people up. And once somebody has been deemed that they are fully trained, we will, of course, allow them to go on these machines. But currently, kind of the default is we allow basically extremely well-trained people. And the final kind of point I will make when I say thank you is that Dr. Omar has instructed me, as like people say, well, what does it take to get on this? You know, is this some kind of inside thing or something? It's nothing like that. Only question I need to ask you is, do you have an IBC? Do you have an IRB? If you have either one of those, we can talk. And because we will not do illegal experiments in anywhere in QBRI, as long as you have the IBC, IRB, I don't care what you're bringing in. That's not my call. I will do the specific experiment that I have been tasked to do for you. And that's my last slide. Okay, thank you.
Thank you so much, Dr. PK, for this interesting presentation and talk. Our next speaker is Dr. Shahrayar Khattak, the manager of Stem Cells and Genome Engineering Core. Dr. Shahrayar?
Yes, hi.
The floor is yours, yes.
Hi, thank you. Just need to. Okay, hi everybody. Thanks for joining us. So I'm going to tell you about the Stem Cell and Genome Engineering Core here at QBRI and what we can offer. Basically, we support all the aspects, let's say all the aspects of the pluripotent stem cell projects. So pluripotent stem cells are the cells that can generate all the cell types of the body. So basically, the embryonic stem cells or the induced pluripotent stem cell. So first, there are the three services that we offer are reprogramming, genome engineering, and directed differentiations. So reprogramming is a phenomenon or a process by which you can convert a normal cell or any cell of the body into a pluripotent cell stage. So basically, you can take skin cells, you can take blood cells, and then convert them into an induced pluripotent stem cell, which are normally called like iPS cells. You must have heard about these, and people are using this. And then from those iPS cells, you can make different cells of the body or do other studies. Genome engineering, basically, you must have heard about CRISPR-Cas, and before that was TALENs. So what we can do here in the core is like modify the genome, I would say up to certainty, like almost 100% or more than 90% of the genome can be modified by using these different recombinases or the nucleases that are used in the field. We can do it on any cell lines as well as also on the pluripotent stem cells. So as I mentioned that the pluripotent stem cells can differentiate into different cell types. So we also offer directed differentiation services. Basically, if you would bring your iPS cells or ES cells, like embryonic stem cells or iPS cells, we can generate for you cortical neurons, we can generate brain organoids, and we can also offer pancreatic beta cell to you from those iPS cells. And of course, as PK mentioned, we are happy to work with everybody, internal or external users. So I'm going to go through the reprogramming process first. So basically, there's a nutshell, like an overview. So the process takes around 4 to 6 months. And what we do basically is that we meet the researchers, and there's a sample submission kind of process that Adil will explain in the end. But this is just to give you an overview of like what we are going to do. So once you bring us your somatic cell or any other cell type or blood or whatever, we will extract the cells that need to be converted to iPS cells, basically to be reprogrammed. And then we'll just reprogram them. We will pick up iPS cell colonies, and then we will karyotype them. So karyotyping is a process whereby you look at the different chromosome structures as if they should be correct, and there is no translocation or duplication or deletion has taken place in the chromosomes. Once that is confirmed, we will further characterize them for a pluripotency characterization. So basically, we would make sure that the cell is a bona fide pluripotent stem cell rather than the original somatic cell that was submitted. Once that is done, we will generate data for the pluripotency characterization, and we will deliver you 2 iPS cell lines per each somatic cell. So if you would submit one patient sample, blood sample, we will give you 2 iPS cell lines, as well as all the characterization data and the karyotype data for this for this service. So the next one is the genome engineering service. So basically, if you have a project which requires you to make a reporter line, a GFP reporter line, or any other thing you want to create a mutation, or you want to fix a mutation, or you want to just delete a big region of the genome, so you can come talk to us. We will help you design the strategy. We will help you tell you what are the ins and out of all these things, and what are the advantages and disadvantages of one versus another method, how to do this, achieve your goals. Then once we agree on this, we will design everything for you. We'll design the recombinase, the nucleases, and everything. We will target if the targeting vector is required. If we have to integrate something from outside, we will design that for you, and we will clone it. We will do all your genome engineering needs. We will test the genomic region by PCR and make sure that if you want a heterozygous clone or a homozygous clone, we'll make sure that it is done at that genomic level. Once it is done, we will also characterize if it is an iPS line or an ES line that you wanted us to engineer. We will also do the characterization, the pluripotency characterization, and the karyotypic characterization, just to make sure that the cells are still pluripotent and they have the proper chromosomal architecture. And then we will deliver you the cells, and in this case also we will deliver you 2 independent lines in case of an ES or iPS cell line, and which will be fully sequenced, fully characterized, and karyotyped. Now, as I mentioned, that there are these different characterization services. So basically, we will deliver 2 independent iPS cell lines or 2 independent engineered cell lines per submitted sample. Each of these would have the karyotyping data, a pluripotency flow analysis, and these are just the proteins that we looked at. I will show you an example in the next slide. Or if you want an ICC instead of immunocytochemistry, or if you want a more quantitative, then we will do the flow analysis. Same goes for the 3 germ layers. Basically, as I mentioned, pluripotency is a property of a cell that would make it go into the 3 different germ layers, like mesoderm, endoderm, and ectoderm. So from those 3 germ layers, the whole organism develops. We'll test them for mycoplasma. We will bank them for you. Short tandem repeat analysis, this is just to confirm the identity. This is a special request. If you really need it, we can do this for you. And in case of the engineering projects, we will give you a figure of the engineering scheme, basically. And so all this data above mentioned will be provided to you in a publication quality. So you could just basically copy paste it into your manuscript. So this is an example of a G-banding karyotype, karyogram. So you can see that this is a female. This is a normal chromosome count. So we'll give you one of this. The second thing is the pluripotency ICC. These are the pluripotency markers that we look at. If you want to have immunocytochemistry with nice figures, we will provide you figures like this. So we will have an OCT4, the TRA160, SOX2, and SSCA4. This is just to show you an example that we can do both matrigel and also on the feeder cultures. We can generate both IPS on matrigel and feeder cultures. If you want to go into the flow analysis, we can do flow analysis, and we will provide you these graphs, which will be like the analyzed data. So it will have the single stains. You can see the single stains, and you can see the double stains, as well as you can see the cells that are positive for all four stains. Because all these four markers, we have developed a assay that we can detect all four markers in the same tube using different fluorophores. The next thing is the 3 germ layer differentiations. So basically, as I mentioned, ectoderm, mesoderm, and endoderm. We have 2 approaches to do that in the field. One is the directed differentiation, where you force your ES or IPS cell into a specific lineage. And the other way is to do a spontaneous differentiation, where you just put your ES or IPS cell in a serum-rich media and just let them do what they want to do. In the directed differentiation approach, we will give you the flow analysis. And in the spontaneous differentiation approach, we will give you the immunocytochemistry images. So these all the data that I just showed you, the examples, like this is the quality of data that you will receive if you would have us do reprogramming for you or the engineering for you. Now let's come to the directed differentiations. So in the directed differentiation approaches, we have 3 types of services, as I mentioned in my first slide. So there's cortical neurons, brain organoids, and pancreatic beta cells. So the cortical neurons basically are the neurons in the cortex of the brain. So basically, that's an overview of the protocol that we use. So we start from the IPS cells or ES cells, and we just make these 3D embryonic bodies, and then we make a primitive and definitive neuroectoderm, followed by neural precursor cells. And from these neural precursors, these are similar to neural stem cells. And then from there, we can generate neurons. The whole process is depicted here. So basically, you can take up to a month to get into the neural precursor stage. But then, depending on the experimental needs, we have to mature those neurons from 6 to 8 weeks. So basically, around 3 weeks, they become functional, and you can see good action potential in here. So just an example, yeah, so here is a culture with neurons derived from a healthy donor, a healthy IPS cells, and you can see the dendritic marker MAP2, and the whole culture is almost filled with MAP2 positive dendritic neurons. Also, these neurons are functional, so electrophysiology is also one of the things that has been established at QBRI by Dr. Park's lab. So you can see that these neurons can generate spontaneous action potential as well as evoked action potential, and these action potentials can be silenced by using TTX, confirming that these are really bona fide neurons and having a normal action potential. Let's come into brain organoids. These are like, you must have heard about these organoids. It's like relatively newer things that these are 3D structures reminiscent of kind of a mini organ. So here is the how it is done. So you take the IPS cells or ES cells, and here you make the EBs, but the embryonic bodies, but these are basically a fixed number of cells. So you get uniform EBs, and then you embed them in a matrigel drop, and then you put them in a spinner bioreactor. I'll show you a picture of this in my next slides. And then you can, after 15 days, then you can keep them in this circulating spinner for maybe a year. And then you can analyze it for your experiment. So here is an example, 2 examples of a day 30 organoid. So you can see that they have like all these neuroepithelia and neuroectoderm forming on the edges. And when you basically cross-section this, so this is a later day organoid, which is day 50, because they need time to mature. So you can see that appreciate the PAC6, which is forebrain and neural stem cell marker, the intermediate marker, and the phosphohistone 3, which is like the cells dividing on the apical surface. So I just wanted to give you an example of this just to show you that these are like really brain organoids, and they represent like the right pattern in what is expected of a organoid. And they also have the cortical layers. Basically, you can see like the cortex has basically 6 layers. So you can see like the deep layer markers are there, the upper layer markers are there, and as I mentioned, like the intermediate progenitors and the stem cell and the forebrain progenitors are also the markers are also expressed. So this is an example of a day 40, and this is a day 60 organoid when they have more matured a little bit further. So the last thing is the pancreatic beta cells. Basically, so this is the spinner that we use for our organoids also. We grow them in here. You can also use a shaker, but I mean, this is much more economical, and you can grow much more organoids in this setup than on a shaker on a 6-well plate. So this here is a 30 ml biot. It comes in 2 sizes. It's 100 ml and a 30 ml. And this one is called a spinner flask. I mean, this one also a spinner flask, but it's normally used called a biot. So here, this is a 300 ml biot. So basically what we do, we generate our pancreatic beta cells from human ES or iPS cells in these such systems. So if we need more cells, we go up to here. We can seed up to like around 180 million cells. And once in 3 days, they reach around 450 to 500 million cells. And then we start the differentiations in here. And like it's 10 times lower. So we start with 18 million and we get up to 40 to 50 million cells. Currently, we have the expertise that what we provide. The service is only based on the human embryonic stem cell line, Harvard University embryonic stem cell line 8. So we provide the beta cells using this one. In future, we'll provide the patient-specific pancreatic beta cells as well. So just a quick overview of the protocol. Protocol is divided into 6 stages.
Have a couple of minutes.
Yeah. Okay. Okay. Thank you. So basically, we go up to stage 6, and it takes around 30 days. And I'll just quickly go through. Like we have a lot of quality controls based on morphology, also based on flow cytometry. And I just wanted to show you that there is no difference if we are using a 300 ml versus a 30 ml. Only the difference is in the size of the clusters, but the functionality is all the same. So here is the pluripotent stem cell stage, is stage 1. Now we have at the another stage, which is the stage 3. And here you see the same kind of like morphology, but different sizes. And this is the pancreatic beta cells that we generate at the end. These are the clusters. So here it's bigger and here smaller, but both are functionally active and they are fine. So what data what you get from this is like basically the flow graphs that we will give you. And this is for different stages. So basically the endoderm stage, the progenitor stage, and then at the end stage where we can see the NKX 6.1 and C-peptide positive cells, which are insulin, which is a byproduct of insulin. So in summary, basically in reprogramming, we can reprogram any cell type into an iPS cells. We will deliver you 2 iPS cell lines per submitted sample with all the pluripotency and karyotype data. And we will give you a 5-vial cryopreserved bank for each cell line. Genome engineering, we can do any type of genome engineering, what you want. And then we will also, like if it is a iPS or ES cell line, we will also provide you with the characterization data, and we will give you a 5-line bank. The characterization data, for both these things were listed earlier, and so this is the one. So I won't go into the detail. So this is the pluripotency, karyotyping, 3 germ layers, and this is enough for a publication. So you don't really need extra. Like you might find a lot of other things in the literature. Oh, they did this, they did this, they did this. But believe me, I mean, we've been doing it for a very long time, and I've published only with this one, and it is accepted in really high-profile journals. Directed differentiations, we have cortical neurons from a control iPS line, and we can also take your ES or iPS cell line and generate cortical neurons. We'll give you a live culture of 24 well plate. of 6-week-old mature neurons. And if you have submitted your own cell line, we will also give you neuroprecursor cell bank, cryopreserved bank. Brain organoids, right now we are doing a healthy control line, and we can try yours, but it might require a little bit of troubleshooting. And we can generate up to, we'll give you or deliver up to 30 organoids at user requested age. And beta cells, we are currently doing the US8 cell line, and we will give you a 14-day mature stage 6 beta clusters, which would have up to 30% of NKX6 C-peptide double positive cells. And we will also provide you with the flow data for stage 2, stage 4, and stage 6. So thank you very much for your attention.
Thank you, Dr. Shahrayar, for this insightful presentation to the stem cell core. And now our next speaker is Dr. Houari Abdeslam, the manager of the proteomics core. Dr. Houari, the floor is yours. Dr. Houari, can you unmute yourself? Dr. Houari, can you unmute? You're still muted. Yes.
Hello, can you hear me?
Yes, I can hear you. And can you put your slideshow on the yes.
Thank you, Hawla, for the introduction. And I would like also to thank the communications team for organizing this event. Hello, everyone, and welcome to this core facility webinar. I will be giving you a presentation on the proteomics core facility. I'll be talking about its state-of-the-art technologies and services. One of QBRI's goals is to support protein-based research activities and biomarkers discoveries towards studying human diseases that are the main focus of QBRI research programs. To this end, we have built and established a new proteomics core facility that is set to benefit from technology transfers from two world-leading companies in the field of proteomics: the Swedish Olink company and the Singapore-based Cengenics company. In 2019, QBRI has executed agreements with both companies to provide Olink and Cengenics services in Qatar and abroad. This year, Proteomics Core has been certified by both companies to be able to use their technologies and provide services. Now, something about the research scope of the Proteomics Core. Discovered biomarkers are set to provide insight into the disease progression from early to late stages, understand the biology and pathology of the disease, and discover new drugs. This is very important to precision medicine approaches where diagnostic, therapeutics, and monitoring treatments are personalized to specific patients, leading to a better management of the disease. Now I will talk about different services that are provided by the Proteomics Core. First, Olink technology is a multiplex immunoassay that can target over 1,200 biomarkers, split in different panels. Each panel is a 92-plex. The technology uses qPCR technique, and for that, Biomark HD from Fluidigm is the most suitable equipment for Olink technology. Olink technology offers a range of protein biomarkers, which includes 15 different panels. Each focuses on specific area of a disease or key biological process. It gives you the choice and flexibility on what to explore. As you can see here, we have cardiovascular panels, oncology, even development, organ damage, neurology, and so on and so forth. Next, I would like to show you a video on how the Olink technology works. So the technology calls proximity extension assay that can use different biological fluids such as serum, plasma, CSF, and other biological fluids. This needs only 1 microliter of blood to measure protein profile in patient samples. Samples are incubated with the PEA probes. Matched paired antibodies carrying unique DNA tag bind to the respective proteins in the sample. When the matched DNA are brought to close proximity, they can hybridize In case of non-specific binding, the DNA cannot hybridize. Only the correctly hybridized tags can be extended to an amplicon with the unique sequence for each protein. Olink targets using qPCR enable digital readout of 92 biomarkers simultaneously. And this is in the 96 samples kit. So the number of qPCR cycles is related to the concentration of the protein in the samples. Statistical analysis tools will provide robust identification of individual protein profiles unique as a fingerprint. And this will lead to stratification of subjects into protein signatures. Next, I would like to present the second platform of biomarker discovery. It's called Sengenics. It's another biomarker discovery technology that is based on autoantibodies detection in biological fluids such as serum, plasma, urine, etc. 1,600 proteins are immobilized on a slide. The autoantibodies in the serum binds to the proteins. Then those antibodies are detected by fluorescently labeled secondary antibodies and then scanned in a microarray scanner. The one that we use is from Agilent, this one on the right. This is an overview on Sengenics technology. It's called the CREX protein folding technology. Unlike misfolded proteins, correctly folded proteins are biotinylated and immobilized on slide microarray. The technology targets autoantibodies that were raised against full-length, correctly folded, and functional proteins that ensure high specificity and sensitivity. In this slide, I'm showing the microarray products, Sengenics products. So Sengenics offers diverse microarray products. The main one is an exploratory array called Immunome protein array. It can detect up to 1,600 autoantibodies. The antigens array covers transcription factors, signaling molecules, inflammatory molecules, etc. Sengenics technology can be used to identify autoantibodies biomarkers for early diagnosis of cancer, infectious diseases, autoimmune diseases, and neurodegenerative diseases as well. It can lead to discovery of therapeutics, autoantibodies, and also enable better understanding of disease pathology and characterization. Beside Olink and Sengenics technologies, the proteomics core offer other services that are also biomarker discovery driven, offering single and multiplex assays. So the Simoa HD1 DX analyzer from Quanterix is a fully automated digital immunoassay instrument capable of analyzing samples using a single molecule array technology. It delivers ultra-sensitive measurements of proteins at femtomolar concentrations, offering a roughly 1,000-fold improvement in sensitivity. Quanterix offer different kits, which are mainly inflammatory and neurological biomarkers, as either single or multiplex assay. Next, the MSD Mesosector S600 is a plate reader designed for single and multiplexing assay of biomarker discovery. The combination of rapid time of 70 seconds per plate and the ability to process both 96 well and 384 well multiplex plates makes the S600 ideal for many demanding research projects. So the MSD has an extensive also catalog of complete assay kits, which are related to cancer, inflammation, metabolism, neurodegeneration, etc. Lastly, the BioPlex 3D from Bio-Rad is the next generation multiplexing platform. It has expanded multiplexing capability. It can measure up to 500 biomarkers. The plates read times is twice as fast as the BioPlex Luminex 200 system. It offers a range of multiplex panel ranging from 48 to 65 plex, including cell signaling biomarkers, cytokines, cancer, diabetes, oncogenesis, and so on and so forth. This was the last slide. And finally, I would like to thank QBRI management for the great support they provided for the establishment of the proteomics core. Here in the right, you have the contact details for anyone who is interested on our services. And thank you all for listening.
Thank you, Dr. Houari, for this informative presentation on the proteomics core. And now our next speaker is Dr. Amani Daher. She's the head of the clinical research core. Dr. Amani, the floor is yours.
Thank you, Khawla. Let me share my presentation. Hello, everyone. Thank you for joining us today. I'm going to give a short presentation for the internal service about the clinical research core here in QBRI. So the clinical research core is a new core at QBRI. Our main mission is to provide an SOP and resources for different biospecimen to support the internal users at QBRI. It is also designed to provide a storage support for samples operated through QBRI. And we are also facilitating the collaboration among internal users for samples sharing. So what do we have? For the core lab, we are in the core equipped with the necessary equipment required for the proper safe handling of the different types of biospecimen from the cabinet, freezers, fridge, the storage system, centrifuge, labeling system. So about like a specification, what do the clinical research core provide for the investigators at QBRI who are involved in a clinical and translational research projects? We support, facilitate the daily research activities. We are providing also a solid practice and create evidence-based SOP for biospecimen collection, processing, and storage. I will be talking about this shortly. And we also providing a digital record in terms of the location, quantity, custody, the history operation are securely stored and managed in the core. So we are doing two things on the site and on the lab. So our roles and responsibilities for sample collection on the clinical site. First, we provide a supportive documents in terms of the preparation of a template, SOP template, sample collection and storage form. Another essential documents as needed for the site, like the lab data, notes, etc. We also provide a study contribution in terms of dedicated staff for recruitment and activities for the specific research project. We also can provide a site selection and communication. We can like support on this with the collaborators. For the samples handling, we have like well-defined rules and regulation of samples handling with the agreement of the PI using the internal service. And we are also following for the sample transfer the procedural guidelines. For the lab, so when the samples received in the core, we do inspection for the samples, review the attached forms and documents. collected. So we ensure that the collected samples with full adherence to the material transfer agreement, for example, the informed consent is there, the IRB. Then we do the process. So currently we provide like sample processing following the established protocol. And the core lab will also work closely with the PI to adjust and validate other sample processing as required. And then we do the storage in a secured freezer in the core lab until further process as required by the investigator. So for the sample release, for the internal user, if they want to inquire samples from the core, the researchers looking to acquire a biospecimen from the biorepository, they may submit a retrieve form. This retrieve form should be filled by the requester and also the project coordinator as well as the head of the core. After the form filled and submitted and the approval then will be the samples will be released to the end user, and then it will have like automatic update in the core inventory. So this is the key documents that we are currently using: the retrieve form, which I talked about, the processing logbook, and the shipment collection form. And this is mostly our service for the internal users for QBRI. Thank you.
Thank you, Dr. Amani. Thank you for this presentation. And last but not least, Mr. Adil Abu Zahri. Mr. Adil is the lead of the information management, and he is overseeing the PPMS workflow and the booking system, supporting all the core facilities. Adil, I will hand over to you for your presentation.
Hi, Hawla. How are you? And thank you for this introduction. Just 1 second, I want to share my... It's not coming, just 1 second. Sorry about this, I have a lot of applications running out. Okay.
Yes, yes.
Yeah. First of all, thank you, Hawla, for this introduction, and thanks for my colleagues about nice presentations that already before me. Now I'm talking about core management system. As explained before, each core has a different methodology and different process, as well as different submission requirement. In this presentation, I'm only explain the main workflow and high-level workflow. It's shared for all cores. If you have a specific requirement or a query, you can ask the core manager, the required core manager. Now, already Hawla introduced me, and this is my mail. Hawla can share it in the Q&A session. Now, in this presentation, I will cover why we use this system, and how will and who will use the system, and how we can use the system and utilize it, and what are the deliverables or results. As mentioned before, we have a lot of services in each core. And CMS or PPMS is a web portal which streamline all the services in a single platform. And this is allow us to submit or request the services from QBRI service catalog, then track the submitted requests which has increased our instruments or core utilization and enable or provision our services online to book and utilize them. Simplify the instruments management, then improving the research outcomes. Who will use the system? Now we have two group of people can use the system: internal users and external users. Internal group of users, it's QBRI users and employee. The external users, it's simply other than QBRI staff. And the difference between these two users, internal user has different schema than the external user in terms of permissions, pricing, invoicing, as well as the service requirement or submission requirement. And each user has a different credential to login. Now, how we can use this system? As I mentioned before, I will share the high-level workflow, which has only the main steps. First of all, consult. Ask the requester or ask the core managers to give you the right services that you have to request. Then log into the system, submit your request, then find your result. That means first consult, then log in, then submit your request, then find your result. Now, how we can use the system and how we can access the system. First of all, it support all kinds of browsers, and you can use your handheld device to access this. BBMS URL is bbms.eu/QBRI. Then this is the landing page. You will choose any of the calls, then proceed with the logins. As we mentioned before, we have two kinds of logins, internal users and external users. If you don't have this credential, you can go you can go through account creation request here form. Then you can fill the first name, last name, phone, and your email and preferred password. Then select your group that belong to. Maybe sometimes there is no group for you. You can create a group from my group is not listed in the list, is not listed or is not in the list. You can create your group, then submit your form for approval. After that, you can proceed with your requests. After submission, your request, then approval, or after request, your service and approval. And after your data has been proceed or sample has been proceed, it's come the results or an email, automatic email will generate and sent to you as a completion email, inform you your result is ready and you can collect it. In this case, you have to contact the core manager to discuss about this receive. This is all. This is a high-level workflow. If you have any question, please ask. If you need more clarification or if you feel you want more hands-on training, we can go with Khawla. And that's it. Thank you, Khawla.
Thank you very much, Adil. First of all, I'd like to thank all the speakers for their presentation and time, and I would like to thank all the attendees for joining us today. Now, let's look over the questions. But before that, I would like, I have my question myself to address to Dr. Richard. Unmute. So from my experience in genomics, I know that bioinformatics support is a very essential complement. My question is how to actually, how can external researchers have access to our available bioinformatics service? Can you give them an overview? How can they reach the available resources we have?
Well, primarily anything that they do that requires going through the genomics core or proteomics core, they would just follow the same pathways that they would use for those. If there's anything specific that doesn't use those pipelines, they can feel free to contact me directly, because there isn't a specific entry for bioinformatics, and we can discuss their needs and see what we can do to help them.
Okay.
Yeah.
So the first question I have here, it is not addressed to a certain, but someone can take this one. I appreciate an overview of the regulatory requirements for establishing the course, if possible, before digging deep. Like he wants like the main requirement for establishing the course. Can someone answer? Gerald, can you give us an overview? How can we establish a core facility or the regulatory requirements?
You were asking me?
It was not addressed to anyone. It's a broad question for any core
Sharia,
user.
I think Sharia
Yes.
has some comments on this, right?
All right. Okay. Yes, please, Dr. Sharia.
So basically, when I can speak for my own core mostly, like of course you need to have the proper permissions to have the level of sample level, like biological safety level 1, 2, 3, whatever core is supposed to do. So basically, like for us, like we have a biological safety level 2. Also, on the other hand, if you are using proprietary technology or which would kind of like overstep on a patent or something like this, you need to get permissions from those. So for example, in my case, like we generate iPS cells. Now the iPS cell patent is owned by Academia Japan. So we, before establishing a core here or before our like service providing, we basically ask the Academia Japan for permission to that if we can do this on a cost recovery basis and not on a profit basis. So basically, then we get a permission from them that, okay, so Academia Japan has allowed most of the research institutions and most of the governmental and universities to use these IPS generation as a core service based on cost recovery, and they don't need royalties from that. So basically, these are some of the things that you really need to think if you want to open a core. So in terms of like, so it just depends on what type of a core you want to open. And then if there are like some infringement on the patents, or if you need to get permissions. So this is my take on it.
Okay.
May I jump into here, because I think I totally would agree what Sharia said, and this is completely, I think, independent or dependent on the core you are having. I just wanted to jump in because I see another question in the chat that might relate to that from Siet. We're asking one of the questions if flow cytometry was used to do any COVID-19 sample here. So together with the question from before, no, not in our lab, because our labs are not at all certified to do infectious virus work, for instance. So we wouldn't be allowed to do that in our labs. As Sharia said, there are different biosafety levels. If you do that kind of work, virus that is infectious, obviously, you need to have, I think it's biosafety level 3 at least, or 4, but it's specific labs that are closed, that have a lower, no, higher air pressure and things like that. So those labs are out there in the research world. And specifically now back to the question, COVID-19 samples have been analyzed by flow cytometry not in our lab, because we are not allowed to do that here. Yeah. So our lab here is a research lab, and as I mentioned in my presentation as well, we have one cell sorter that is in a biosafety level 2 cabinet, but that wouldn't allow us to do any infectious or let's say more tricky material even. Yeah. But there are other labs that are specialized in this. And back to the main question about regulatory requirements, this depends highly on what level of lab you are asking for. So for the flow cytometry lab here, as far as I can tell, there are no very specific regulatory requirements, except the ones, to run a lab, to have all the safety, biosafety level requirements, and so on.
Thank you. Thank you.
Yeah, and coming back to the COVID-19, sorry, Khaled, just
yes, please go ahead, Dr. Shahab.
Yeah. So coming back to COVID-19, so we did not do any flow analysis, but also like Khaled and others have been involved in processing like samples to run diagnostic tests, and we have been helping HMC with that a lot. And those were like all the samples were deactivated, like inactivated virus. So you have procedures to inactivate the virus before it can be taken in. And Khalid was the point on it, and Khalid actually developed the PCRs and the RT-PCRs for that, as well as like I think Huari is also working on something like with the diagnostics aspects of things. So we were involved in the COVID, but not directly in the flow cytometry.
Okay, there is a question for Dr. Gerard from Rajivan Narayanan from Oxford University. Very interesting talk. A question concerning flow cytometry. Suppose if I have a brain slice stained for neurons with a neuron marker, how we can estimate the total neurons in that given tissue sample? Would that be possible? If yes, how long this will take for measuring from a 2 millimeter into and 100 micrometer thick tissue?
Wow, okay. First of all, when I hear the question, I would say, okay, flow cytometry can do a lot, but not, let's say, quantifying cells from a slice of a microscopic slice, because one of the prerequisites for flow cytometry is you need single cells in some liquid, in suspension. So that's the general point. I cannot pass a slice of a brain through my flow cytometer. That wouldn't work. There are microscopic techniques that can do that, in fact, quite quickly as well. If the cells are stained in a way that's also automized, as far as I know, that is possible. Now, if you would have this situation and we would go to a flow cytometer, I can imagine you can extract the specific cells in a way. You can lyse the tissue around the cells. There are methods that you can, let's say, it's basically like grinding meat or something as well. You can destroy the tissue so that you have your cells in suspension, in some solution, PBS, whatsoever, and then I could pass them through the flow cytometer. So if those neuronal cells are expressing a fluorescent protein, for instance, or if they are stained in a way, we could do that. It's always a little bit tricky because what you will end up is with a soup of liquid with all kind of material in it and the cells. So we need to find a way, and this is then where optimization and troubleshooting comes into account. We need for sure, if I think about it, I would imagine for sure we need to filter the sample very well and find an optimized way of treating the sample, digesting the material that is not of interest, filtering the sample so that we are left with mainly the cells of interest in liquid. And then once we are there, we could, in fact, count those cells very quickly with a flow cytometer. That's done, as said, that's thousands of cells can be analyzed in a second. Maybe with such large cells, it's a bit slower, but we could definitely, I would say, hundreds of cells can be counted and analyzed in a second or so. So that wouldn't take then very long, but it's a kind of tricky protocol. Generally spoken, in flow cytometry, everything that is tissue and comes from solid tissue, it's more tricky than if it's blood or water or milk or whatever liquid. But it's possible.
Okay, thank you, Dr. Joel. There is a question addressed to Dr. Shahrayar, stating you showed the cell lines transforming into neurons with dendrites, also with spontaneous and evoked action potentials. My question is, these cortical neurons express any typical type of cortical axon morphologies as well? These cortical express any typical type of cortical axon morphologies? That's the question.
Yes. Okay. So yeah, they do. And if I can share this my presentation, that would be. If I have the sharing, I can basically show you the. So what we did is did QRT PCRs of all the cortical markers, the neuronal markers, and different brain regions, and also immunofluorescence on those neurons, just to show that they were really like bona fide cortical neurons, as well as like the morphology wise, we also checked for different like dendritic markers and like spine markers and all those things. So they do show morphology of those. But for the sake of time, I just like showed you that, okay, yeah, we have dendrites, we have axons, we have PSD95, we have VBRUT1, we have GABAergic, we have
Dr. Shahrayar, please feel free to share if you like now.
Yes.
So basically, this is a single cell QRT PCR using a biomark fluidigm. So basically, what we did is we took live neurons without any. Once these are generated, we just took live neurons and just put them in a well of a 96 well plate, and then used all these primer pairs and did QRT PCR, like basically on a single level. So basically, there were like 48 genes on a single cell transcript, just to see which type of a neuron did we make, which area of the brain it is representing, and what is there. So like basically the simple neuronal markers, if you see like NCAM, MAP2, and DCX, we see that there's more than 80% are neurons. So basically there is most of the protocol, like they generated neurons, and also like different forebrain markers. These are the cortical layers, as I mentioned, like the 6 cortical layers are there. We have some of the calcium relators. So we have the excitatory and the inhibitory neurons here. So I mean, they have everything. So basically, it's a mixed population, and the idea was to generate cortical neurons, as we know that the cortex contains most of the neurons of the human brain. So this is the idea. I hope this answers the question.
Okay. Thank you, Dr. Shahrayar. There is a question for Dr. Houari. Explain the differences between the multiplex system, Meso, Luminix, etc. How would you choose which platform to use? Dr. Houari?
Yes, so this is depending on different factors, depending on what you are looking for. So if you want to do an exploratory study, like you want to screen your, up to 1,200 proteins, you can go for Olink. And of course, in Olink, you can choose specific panels. Like if you are studying neurology, so you can choose neurology. If you are studying metabolism, you can go for metabolism. And then for the transgenics, it's different. So it's autoantibodies. So it means that you will detect the antibodies that they are present in the samples that are raised against, proteins. And the other equipment or the other technologies that we offer, it's also depend on what you are looking for. So everyone is different. For example, for the Simoa, if you want to look for a molecule exactly at the very early stages of the disease, how it's progress. So some molecules which are expressed at a very, very low level, so you can go ahead with the Simoa because it's ultra sensitive. It can detect the molecules at the femtomolar concentration. I think, yeah.
Thank you, Dr. Houari. There is a question, I think, addressed to all the core facilities managers. Is HBKU colleges considered internal users for service requests? From Dr. Hamish McKay.
I think only QBRI.
Okay.
But they are. Yeah, we can discuss this with the management if the HBK are included as internal. But as far as I know, I mean, internal they are QBRI, and all external they are all the other QF entities like Cornell, HBKU. I don't know. Let's ask the others. I'm not sure about this. So maybe the other core managers.
We can answer his question later on via email for sure. There is a question to Dr. Khaled regarding the nanostring.
Yes.
Can you elaborate on the application that are done on the nanostring string platform?
Yeah, for nanostring, there is many applications. There is for gene expression analysis, microRNA expression analysis, non-coding RNA, and also fusion gene analysis, SNP. There is a copy number variation, and also there is protein and phosphoproteins. The advantage with nanostring that we can use challenging samples like FFP samples. But if, for example, gene expression, there is many panels. There is for immuno panels, there is neuro panels, there is tumor panels, there is many panels. I can advise the person who asked this question to go through the nanostring website, and he can see all panels for different applications. There is a lot. Yeah.
Okay. There is another question for you, Dr. Khaled, regarding the single-cell sequencing.
Yeah.
Do you first the question? Do you use HiSeq?
Yeah,
as the geometry
for sequencing. For sequencing, yes. Yeah. We are using HiSeq 2500, and yeah.
And does the user need to use the complete 8 lanes?
No, the HiSeq 2500, we have rapid run. Look, rapid run, we can load, there is only 2 lines, and we can load up to 16 samples of single cells. So 4 per line, 4 or 5 samples in 1 line. Look, so it will be 8, maximum 10.
Okay, there is, I think here, yes. I don't know that the name is not. In genomic technology, do you do ATAC ATAC-seq as well in our genomic core?
ATAC. This is for chromatin accessibility for transposon normally. We never did it, but it's not complicated normally. And this is with transposon technology. So yeah, normally there is some kits that we can use for that, but it's feasible. We can do it.
There is another question, but I don't. I mean, it's from an Oxford guest. For the core facility, are they planning to bring micro CT and release system in the near future? I don't know for which micro CT. Is it for Gerald or?
If it's for me, no, it's not planned for the near future. No.
Yeah, it did not. All right. Yeah, there is a question for Dr. Richard. How do you tackle the single-cell sequencing analysis? I mean, is there a workflow? What is the workflow for the single-cell sequencing?
The single-cell sequencing. So, as Dr. Khalid mentioned during his his talk, we have things set up to do the conversion to the standard FASTQ files that you would get out with anything else. At the moment, we don't have a pipeline down a further analysis set up, but if people are interested in that, that's something we can see about setting up.
Okay, thank you, Richard. From ABM Sharif, I'm working on cell signaling. This is for Dr. Gerald. This question is for Dr. Gerald. I'm working on cell signaling and cell culture, basically plant cell by hormone application. So my question is, what type of nanoparticles are responsible for rapid signaling?
Okay, I'm afraid I can't answer that question. I don't really know, especially in plant cells. What I know, however, is yes, these nanoparticles deriving from cells, those exosomal vesicles or nanoparticles, however they are called, that this is a field since a couple of years, I think, that is really emerging, and people start to see that those particles and vesicles are really like communicators between cells and out for cells to communicate with each other. Now, don't ask me what particles exactly do what. I have no idea, and I don't know if anybody really understands that already at the moment, that as specifically. But that's, I think, the reason why this is an emerging field right now, that people are getting into this and want to understand. Maybe somebody else of the QBRI team here can even comment on this, because maybe somebody is using those particles already, or has characterized some specific particles already. For me, I wouldn't be able to answer that in a more specific way now. I know there is a big role in this. In plant cells, I don't know at all. Maybe there it's different, but I guess it's similar than in animal cells or in human cells as well. But I know it's a field that is emerging, and people are very interested in what exactly those vesicles do and which vesicles do what. I couldn't answer that at the moment.
Okay. I think one other question to Dr. Houari. How many targets can be detected by the Olink platform?
Can you hear me? Hi.
Yes, I can hear you, yes.
How many biomarkers can be detected?
Yes.
Yes, it's so we have it's 1,164 using the target panels, which we do have here in our labs. So the target panels, they are 15 panels. You have oncology panel, immunology panel. So there are 15 panels which cover different diseases. Every panel targets 92 biomarkers. And there have been recently a new technology which was launched by Olink. It used NGS technology, which can detect, more 1,450 biomarkers. But right now we don't have the equipment for it, so we are using the qPCR target panel with the Fluidigm equipment.
Thank you.
And if they need any more information, please, they feel free. They can send me email, call me, and I will be more than happy to assist them with all their questions.
Yes, thank you, Dr. Houari. I think we've covered all the questions that were posted, and if there are any questions, please feel free to send it to the call manager or to us. And in the interest of time, I'd like to conclude today's webinar by thanking everyone for attending. Lastly, I would like to thank Mrs. Asma, Mr. Omar, and Ms. Reem, that are the team that are running the show behind the scenes. And finally, stay tuned for more informative web seminars from QBRI. Thank you for all. Stay safe, and goodbye. Have a good day. Thank you.
Thank you.